so much, Maria Carmen, Josiane. I want to congratulate you and the rest of the team for organizing this free access global initiative that educates so many people and students and the future scientists of the microbiome. So it's my pleasure to be here, and I'm going to talk about early assembly and effect of the environment and birth mode. So first I want to disclose, I have 2 patents that are assigned to universities, University of Puerto Rico and NYU, before I move ahead. So I'd like to start just showing that in utero, the fetuses of all these species of mammals that you can see here develop without germs. This is a sterile environment. These fetuses are receiving chemical signals from the microbiome of the mothers, but they have never seen live bacteria yet, and which happens during birth. Once the sac that holds the baby breaks, or we call that breaking waters, romper fuentes in Spanish, then that's the first time in the life of that baby when they encounter live bacteria. And bacteria in the birth canal of mammals is highly relevant to early development, and is typically important members of that inoculum are boosted by milk, by the milk that the baby will drink. It is also important to note what is the primordial microbiota sources in vertebrates, and to note that genitals and the intestine exit, the anus, in all vertebrates are adjacent. So on the left we have animals that have a cloaca, meaning that's the only canal where they urinate, defecate, and reproduce. And that's the case for reptiles and birds, but also special group of mammals called the monotremes that are egg layers. On the right-hand side, across the span of vertebrates, from fish to other animals, including humans, we have the design where we can see that independently of the urination canal, the defecation is always next to the reproductive canal. And that's the case also in humans. So the inoculum, the microbial inoculum from these sites is really in the region of exposure of the babies that are being born from these mammals. So feces is an important component. Here is showing the very interesting anatomy of the monotremes with only one channel in females or males, where they reproduce or defecate. The intestine is connected there, urinate, etc. In males, even more interesting, the penis is inside that tube. But this is a really exceptional design of nature. The rest of the animals have their join in the vicinity, the reproductive and the fecal, and anywhere in the animal, the urination. So feces and reproduction go together. Once that happens and a baby is born, nature has, the way nature has designed mammals is that these babies will not eat a diet from outside. They will feed on a secretion of their mothers. That's the natural way. This microbiome on these strictly lactating babies is developing under the influence of the maternal milk. And that's a very complex substance that not only provides food for the baby, but also for the baby's microbes and immune molecules and hormones. And then the natural way is that baby, when the baby has developed enough, the brain, locomotion, sensorial, communication, the baby now starts to demand, grab things to the mouth, and this happens at 5.5 to 6 months, then the baby becomes a very deep explorer of nature. And this means a lot of exposure to non-human microbes, mostly soil microbes, environmental microbes. So in our urban lifestyle, in which every society in the world is converging to, traditional lifestyles that were diverse before are now converging into a single urban lifestyle that is the same anywhere. And this is happening, nobody's controlling it, it's just happening and it's unavoidable. What is happening also is that there are a lot of stressors that impair the transmission of microbes between mothers and their babies, and further later in life, the microbiome assembly of the baby. So we have antibiotics during labor, even if it's vaginal. 1 in 3 mothers in the US gets antibiotics. Then we have C-sections. 1 mother in 3 has a C-section. Oftentimes we feed formula to babies and not maternal milk. And then babies get antibiotics, or mothers get antibiotics from pregnancy. That also perturbs the microbiota of the mother and the baby. In average, in the US, babies get 1 course in their first year of life, 3 courses after 3 years of life. Then we use technology that also separates moms and babies, like these trolleys, but also babies typically in urban societies sleep separated from the mothers in very clean rooms in a setting that has very little of nature. So both the early human exposures to mother and family members are perturbed, but also the environmental secondary exposure is perturbed. So all of these compounded effects are reflected in the microbiota of the kids as they develop. And this is a challenge because as we urbanize, there is an association between being able to very effectively control infectious diseases, as we do in urban societies. But instead of gaining health, we are really trading diseases. We are changing infectious diseases for diseases that are of immune nature and that are of early origin. And we know microbes modulate the education of the immune system in early life. And we think that microbes are contributing to the genesis of the rocketing in these diseases that have in common high inflammatory responses: asthma, type 1 diabetes, multiple sclerosis, Crohn's diseases, and more, obesity, other metabolic diseases. So for these diseases, there are 2 hypotheses right now, and both have to do with microbes. The first one had to do with environmental microbes, and is the hygiene hypothesis. The hygiene hypothesis states that if kids are raised in farms and in contact with environment, with natural environments and animal exposure, they have less asthma and allergies. The second hypothesis is the human microbiome disappearing hypothesis. And it states that is really the lack of the impairment in transmission of human adapted microbes that is educating the immune system badly and therefore affecting regulatory pathways of immune responses. And I think both are important. And there is evidence that indicates that both hypotheses have sound grounds. So we know that C-section alters the microbiome of babies. Not surprising, these babies are born sterile, taken sterile into the air of an operating room that is full of mostly skin bacteria. And this graph shows the trajectory on the PC1, on the principal coordinate 1, of the microbiota of babies vaginally born in blue or born by C-section in red. You can see the trajectory is parallel but separated, especially after the first month of life and until about a year and a half. And so we thought of restoring, and the first idea was, well, can we restore by giving babies the vaginal fluids of their mothers? And can we see if the microbiota of those babies normalize? So we did that experiment. It's an ecological experiment. We published a very small pilot, and then in 2021, we finished the big pilot in which Maria Carmen collaborated, and teams with from Puerto Rico and Chile as well. So we had 97 vaginally born babies as a reference to compare 49 C-section babies without seeding and 28 babies that were born by C-section but were exposed to the vaginal fluids of their mothers at birth. And what we found using a very elegant method developed at Rob Knight's lab is that when you compare the blue babies, which are the references, to the red babies, which are born by C-section, the C-section babies have a clearly distinctive trajectory. But if they were seeded, their trajectory was intermediate between the normal and the C-section. Intermediate for the feces. For the mouth, the green trajectory highly normalized to the blue babies born by C-section. And for the skin, it was probably the place where it was easiest to see the differences that C-section babies have. Unless they are seeded, they greatly normalize very well. So we can tell that at least partially in the feces and very well in other body sites, if you expose babies that are born by C-section to vaginal fluids of their mothers, that remember, the area contains also fecal bacteria. And we know that we are now studying that how the vaginal microbiota of mothers giving birth, it's also colonized by oral and skin bacteria. So it seems like the niche is opening to bacteria from other body sites, which would make sense because these babies that are being born have the different body sites, the epithelia that will be colonized. And mom is providing the pioneer inoculum, the first one. So then we said, well, so right now the big trial to look at babies' health is ongoing. It's a 5-year trial where now the question is not only microbiome, but also are we protecting the kids from excessive fast growth and from asthma markers. Now, what we decided to do is how different is the poop from babies that are born vaginally and the babies that are born by C-section? How much perturbance or how different it is, the C-section baby's intestinal content. So to answer that question, we put the feces from the human babies into germ-free mice and look what happened. So from the human inoculum, we could see the differences, and this is showing, this is a Venn diagram of exclusive or shared ASVs. This is 16S sequencing. And on the right-hand side, you can see the much higher Enterobacteriaceae in the C-section inoculum and the much higher one type of bifido in the vaginal inoculum. You can also see other differences like bacteroides. Bacteroides is a very good marker of being born vaginally. It's almost absent in the C-section babies. These babies are 3 months old. When we look at the alpha diversity of the adult mice, the mothers that were gavage, we can see that the alpha diversity in the gut of these mice was higher if they received the vaginal delivery inoculum, the human baby inoculum from vaginally delivered babies than from the C-section. So there is a higher diversity of bacteria from humans engraft in the adult mice, in the mothers. And then this is the fecal beta diversity in adults at 4 weeks for post-gavage. And you can see the beta diversity. You can see the initial pools, humans, and then how the mice select differently, but very differently between the two groups of feces in the inoculum by all of the metrics of beta diversity. We know what bacteria were engrafted from the vaginal delivery inoculum and not from the C-section, and what are the unique ASVs, and then what bacteria engrafted from the C-section inoculum as well. And we know that more bacteria engrafted from the VD than more than half from the bacteria in the C-section feces did not engraft, whereas in the vaginal delivery group, 30% did not engraft and 70% engrafted. So there is a better engraftment from the inoculum of the feces of babies that were vaginally born. There was also a very different picture of the co-abundance network of transferred bacteria into the mice. This shows a positive correlation between different taxa in the vaginal inoculum versus the C-section inoculum. So very different pattern of engraftment between the two, very different community and correlations. We also look at the colon metabolites, and they segregate apart. So the metabolites that are present in the gut of babies born by C-section is different from metabolites from babies, 3-month-old babies that were vaginally born. And the mice are special vessels, live vessels, mammals, that are showing in a way they are increasing the differences. When we look at the metabolites, we see a lot of metabolites that are decreased in the C-section groups, and a couple of them are increased. We still don't know what is the physiological implication of these, but we are characterizing the babies the best we can to understand what is different in the babies that are born by C-section. We look at also at the ileal gene expression of the mice that were receiving human feces, baby feces from babies discordant to birth mode. And we also find differences in the ileal gene expression of these mice. These mice, by the way, were gavage when the mother was gavage when the babies were 3 days old. So they received the gavage via the mother. We also study the T-cell profiles, responses of the transplanted mice at week 6. And we observe differences in the CD4+. We observe better Th17 responses in the mice that were given the vaginal birth inoculum, lower Th1, lower pro-inflammatory, higher Tregs response in the mice. So we are already observing phenotypes, immune phenotypes in the mice that are differ by the inoculum the mice received. So we think there is functional relevance, that is bacteria in the gut are responsible of modulating immune responses and immune cytokine profiles in the mice that receive the two different inocula. They have different microbiota, different metabolites, and the mice on the host side, the mice are showing different immune responses. So our urban life has changed the exposures to microbes, not only by the stressors that are like C-section or antibiotics or in the early life that impair human-to-human transmission. But we have also impaired exposure to non-human microbes, as I said before. So one study we did with Joanne Kombelik when she's a midwife who did the PhD in my lab, and she delivers babies at home or in hospital with a team of midwives. These are very low medicalized, more natural births. And we did an experiment in the area where she works in upstate New York, in an area of very homogeneous social class, mostly high middle class white women who are thin and by, organic and etc. So very homogeneous group of women that were delivering with midwives at home or in the hospital. And I was very pessimistic with Joanne wanting to do this, because I said, you really won't have, I don't expect to see any differences, because these are all vaginal deliveries without antibiotics, and it's strictly breastfed kids. So, and from a very homogeneous background, I never expected to see that the babies would have different microbiomes, but they did. So by site, although alpha diversity was not different, we saw differences at 1 month. These are all the data together, but the major differences were after 1 month. They segregated significantly the microbiota of these babies from between home and hospital birth. And when we look at the taxa, we could see that a major group that we also always see increase when there are stressors. They could be antibiotics, or it's Enterobacteriaceae. And we saw that in the hospital, these babies born in the hospital had much higher Enterobacteriaceae than at home, and had lower Bacteroides, which is the marker of the C-section. So this is, again, they were a little perturbed, significantly so, not as much as C-section or antibiotics, but in the same taxa were affected, regardless of the stressor. And this was really a surprise to me, that just the experience of giving birth at home or in the hospital. And what we found is that when we look at the metadata, in the hospital, I don't think it's the air of the hospital. It's not the environment. I think it's the experience. There was a major differences in the responses of the women to, for example, anxiety. Level of pain was higher in the hospital, and therefore they receive more analgesia in the hospital than at home. They wanted to go home, faster. The labors were faster because some of them were induced in the hospital, but not at home. So we realized that the experience is radically different. Even with midwives, the hospital experience is an experience of higher stress, higher pain feeling, higher medicalization, higher induction of labor, anesthesia, and anxiety to go home. So that is fascinating, and it's a field of research that I think needs a lot more. So then we decided, okay, let's see the effect of the environment, and for that we use mice that were born and raised in the farm or in our clean SPF vivarium in the animal facility. The mothers came all from the animal facility. They were bred there and then brought when they were pregnant left in the vivarium or brought to the farm. This is unpublished data. So we could see that the first factor that affects the microbiome development is age. So we are comparing here week 3 in purple, week 7 of development, and week 12. But the second one was facility. And the facility between the vivarium and the farm was a major factor affecting the microbiota development. We also, I'm not going to talk about the treatment here, we gave a treatment as well. But the environment does affect at week 3. You can see the green is the farm, the vivarium, and the yellow is the farm. At week 3, at weaning, they were quite different at week 7 and at week 12. These were mice with their mothers, nothing else changed. So this is the effect of environmental exposures on the engraftment of the mouse microbiota. So this is, again, fascinating because it's showing how the external environmental exposure is changing the way that the mouse bacteria, the host bacteria, is transferred between generations. So in summary, mammals acquire pioneer microbial colonizers during and after birth. Surgical birth alters the microbiome and can be partially normalized by exposure to maternal microbes right after birth. The differences are reflected in the microbiome, but also in metabolites and host immune responses that are associated to the microbiota of babies that are born by C-section versus vaginally. We think that the experience of being born in the hospital or at home have associated effects on the microbiome of the babies. And even as the mouse experiments show, the environment itself is having an effect on the way bacteria from the host is transferred between generations. And with that, I want to thank my lab, which is depicted here. I have 2 postdocs and 5 PhD students and 7 undergrads that work with them. And then Rutgers colleagues, including Marty Blazer, Liping Zhao, that you may know. Gary Wu works on the microbiome. Manny DiCicco works on neurodevelopment. We are working with him also to look at the brain of mice born by C-section. Troy Repke is a nutritionist, also expert in nutrition and behavior in mice. And then the team from the University of California, San Diego, including Rob Knight on the microbiome side, and Peter Dorestein, who on the metabolite side. Collaborators from UPenn, the CHOP, Kyle Bittinger and Dan Scott. And then the team from Venezuela, where I'm still working with them to understand, being born in traditional communities and growing in traditional communities. That's a project that is still ongoing. And with that, I thank you all for your attention, and I'm hoping you have questions for me that I can answer. I'll stop sharing.